my master's thesis, which is image analysis of settlement plates for monitoring marine benthic communities of the Kerguelen Islands. A preliminary study has been done in the laboratory Biogeosciences as a part of the program of the French Polar Institute and in partnership with the Natural Reserve TAAF. So Kerguelen Islands are part of the French Southern and Antarctic lands and are located in the southern Indian Ocean and are situated exactly in the eastward flow of the Antarctic syncopolar current and are situated between two main marine fronts right here. So the coastal areas and its benthic community are likely subject to different water masses from different temperature, which is an important issue in the current climate change context. So from these issues has emerged the PROTEKER program, which is a multidisciplinary observatory of coastal areas around Kerguelen Islands. It's 8-year survey from 2013 by means of settlement plates, colonization process assessment, so which we will be focused on this study. And for the program is coupled as well with abiotic factor continuous measurements as well as molecular analysis. So the main objective of this study is to provide a broadly comprehensive description of structure and composition as well as evolution of macrobenthic communities on settlement plates. And the question of this study is to which extent image analysis allow a thorough survey of benthic communities dynamics at 3 sites of the Kerguelen Islands. How are we going to do that? We're going to be analyzing ecological succession from early stages by means of photoquadrat analysis. So what is ecological succession? On the one hand, we have primary succession, which is the recolonization of organism on newly available bare surface. This way, starting from pioneer, early stage, late stage, and the final hypothetical biotic stage, which is climax. And on the other hand, we have secondary succession, which is this time recolonization, of surface which have been previously dephonated due to disturbances from biotic or biotic origin. So for this study, we're going to focus on primary succession because secondary succession would be more linked to isolated events, and our aim is to establish a local scheme of succession on how it occurs over there. So as ecological succession implies important change in diversity and community structure, we're going to be analyzing alpha diversity, beta turnover through time, as well as we're going to try to investigate morphotype, which are mainly involved in these shifts. Regarding the sampling site, we have chosen 3 sites out of the 8 all reference 1 for the program for experimental design homogeneity. They show different oceanographical feature. Île Haute is situated at the back of the bay, as you can see, is close to river outputs and is therefore more likely subject to variation from terrestrial origin. Île Soum is highly subject to ocean inputs and hydrodynamism in general, and Île Longue is quite physically protected. So for the experimental design, in each monitor site, 8 clay settlement plates have been set up at 10 meters depth in 2013, so this way. And each position plate, well, as a function of the position, the plate will be removed after a different amount of time. So here the red dots is when the plate is collected and subject to molecular analysis. So for example, this position is collected every year, this one is collected every 2 years, and so on. It allows to have replicate of age and years. We have at our disposal photograph of the plates which have been taken yearly, as well as photograph from the adjacent environment from 2019, and the adjacent environment will be our hypothetical climax condition. The photoquadrat analysis has been done through the software PhotoQuad. We have established a morphotype library that we have loaded out inside the software. And we have used the stratify random point method, which is the quadrat is basically divided into 100 subquadrats, and within each quadrat, one point is randomly placed, and at the end, each point is attributed to morphotype in order to obtain coverage data and species richness. Regarding the data analysis, we have first performed a methodological analysis. On the coverage data, we have used the a correspondence analysis in order to get continuous data instead of percentage, which are quite dependent in between each other. Then we have treated this data independently as a function of the age class of each plate. And for each age class, we have performed a multivariate ANOVA to try to detect a significant site or year effect on morphotype CA scores. Then we have calculated alpha diversity indices, so species richness, Shannon-Wiener index, P elusiveness, and that's it. And regarding beta diversity, the analysis has been done through primer. So we have pre-treated the coverage data to the square root. We have performed the Bray-Curtis matrix, cluster analysis, and non-metric multidimensional scaling. And finally, we have performed principal component analysis on morphotype coverage as a function of the community, so each year plate and adjacent environment. So for the first results, for each age class, we have found a significant effect of the site, and for the plates of 2 years old, there is an effect of the year. Therefore, as we want to analyze the ecological succession, which is a long process through time, implying a lot of evolution in community, we have decided to focus only on 3 plates for this preliminary study. For 2 reasons. First of all, to avoid the significant year effect we have just detected, in order to not compare plates of the same age but from different years, because there is an effect of the year. And the second reason is that they are the only plates which have remained untouched since the beginning of the monitoring program. So regarding alpha diversity, the 3 monitoring sites did not display exactly the same pattern in terms of diversity, but for this presentation I decided to gather them just to highlight the main signals which are actually common to these 3 sites. So we see that there is important evolution the first 3 years of the settlement process and less variation the 3 following years, commonly to the 3 indices. Overall, Shannon index and species richness improve through time, and P elusiveness decrease. And species richness is significantly higher in the adjacent environment than the plates in general, and the adjacent environment for the Shannon index is significantly higher than the early settlement plates. For the NMDS, the first thing we can distinguish, it's the 9 quadrats. Yeah, I forgot to say there is 9 quadrats, 9 photographs of adjacent environments, are likely to be more similar to each other than they are from plates. At haute, it seems apart. We have one part with the settlement plates of whatever the stage, and in one part we have adjacent environments. In Ilsum, it seemed to evolve more toward the adjacent environment, but seemed to display different trajectories, as the adjacent environment is highly spread on the vertical axis. And in Ilong, already from the early settlements, it's already really similar to the adjacent environment. And we see that it evolves significantly toward only 2 quadrats of the adjacent environment, but display more evolution the last 3 years than it does for Sum and Haute. As we see, the last 3 years are kind of grouped together. We have performed, as I said, principal component analysis. So we have projected the morphotype on the multidimensional space as a function of the communities as variable vectors. So communities, as I said, it's each year community plus adjacent environment. So, for example, it's just some example of investigation. At long, the bryozoa Beania sp is more likely to belong to 2018 if we have a look at the length of the vector. And broadly, the 2 colonial ascidia, polychaetes spa and Didemnum sp, are more likely to be ubiquitous, and the mussel Olacomia is more likely to belong to the adjacent environment, and perhaps exclusively. There is more analyses which have been done to confirm that pattern through an MDS and bubble plot for each morphotype. And here we have performed a zoom in the PCA at haute. As we see, the variable vectors are really short for early settlements. So we have taken the coordinate which correspond to this position, and we found that the polychaetes P orbis SP is more likely to belong to early settlements. So from that result, we can say that we hypothesize that the increase of diversity indices at early stage of settlement is likely due to the space availability on plates based on other trend on global coverage evolution in settlement plates. According to alpha diversity and beta diversity results, we can cautiously say that the hypothetical climax condition has not been reached even after 6 years colonization process. Successional system more likely display a variety of evolution trajectories towards variety of adjacent environments, like we saw in SUM, seems to have different trajectory instead of one canalized and predictable trajectory. It can happen as well, but it's more likely to happen like this apparently. Common points have been found between sites regarding the morphotype, which are mainly involved in the community structure shifts. And from that, we could establish a simplified scheme of succession model, with the polychaetes P orbis would act as a pioneer. The colonial ascidia Didymnum sp is likely to be ubiquitous. The two bryozoa, Beania and Celaria, would appear at mid-succession, and Polyclinidae gen sp, another colonial ascidia, would appear at the end of the 6 years colonization process succession. On the other hand, this preliminary study has shown as well differences between sites, obviously, as it could be suggested by the preliminary analysis with the site effects, as well as the different oceanographical features of our 3 sites. We hypothesize that the biotic factor may play an important role in these differences regarding colonization process. It could act on the rate of primary colonization. For example, as I said, the local scheme seem to, in terms of morphotype, seem to occur in the 3 sites, but at different rates. For example, it seems to appear some species like Polyclinidae, which is supposed to appear at the end, is likely to occur earlier in long than in haute, for example, which can be due to a biotic factor and of course exposure. And it can be due as well to historical background and site sensitivity to potential previous perturbation, and we would be in the secondary succession case as exposed previously. This preliminary analysis highlight as well several photoquadrat analysis limitation, such as taxonomy and organism identification. In macrobenthic communities, there is small organism which needs to be either open up or subject to molecular analysis in order to be
2 minutes
to be identified to the species level, and a biotic factor analysis for a better interpretation of successional processes. So to which extent image analysis allow a thorough survey of benthic communities dynamics at 3 sites of the Kerguelen Islands? So this method has allowed to identify ecological succession process through 6 years colonization and community structure evolution, and it has also allowed to establish a simplified scheme of succession and on how it occurs over there. But on the other hand, it would be advisable in the future to couple image analysis with other methods in order to strengthen the benthic community survey assessment, identify factors which may be implied abiotic or biotic in differences between sites as well as between plates and adjacent environment, to try to understand why some species even after 6 years cannot thrive on settlement plates but are significantly present on the adjacent environment. Further researches are needed. Thank you very much.